My name's Megan, and I did my gene project on the FOXP2 gene. So, some learning objectives for this presentation are to understand how Mendelian genetics applies to the FOXP2 gene, how mutations in the gene disrupt speech and language, to understand the location and function of the gene, and to understand the function of the FOXP2 gene product. These 4 goals will guide everything that I talk about, and by the end of this presentation, you should have a clear idea of why this gene is so important, especially in relation to human communication. So, starting off with some background, the FOXP2 gene appears across a wide range of species. It's found in humans, primates, birds, fish, various mammals, and various vertebrates. Because of this, scientists often study FOXP2 in different organisms to understand how vocal learning and communication systems evolved. Even though humans have a unique and more complex ability for speech, the basic building blocks controlled by FOXP2 appear in many animals. The gene product of the FOXP2 gene is the FOXP2 protein. It's a member of the four-head box family of transcription factors, and they play important roles in cell growth and embryonic development. This protein is a transcription factor, and it regulates other genes by binding to DNA. It most notably targets the genes CNTNAP2, SRPX2, SCN3A, and DYNAPT1, and it does this by repressing those genes. It often acts as a repressor, but under certain conditions with specific cofactors, it can act as an activator as well. The FOXP2 protein is active in several tissues, including the brain, both before and after birth. Studies suggest that the protein plays a key role in the functioning of synapses, which are the connections between nerve cells where cell-to-cell communication occurs. This protein is specifically important for synaptic plasticity, which is the ability of synapses to change and adapt to experience over time. Synaptic plasticity is necessary for learning and memory. The FOXP2 gene is essential for the normal development of speech and language. Researchers are still working to determine which genes are influenced by the FOXP2 protein and how changes in their activity lead to abnormal speech and language development. The phenotypes of this gene are childhood apraxia of speech, dysarthria, and challenges with articulation, grammar, and fine motor coordination. Symptoms for childhood apraxia include babbling less or making fewer vocal sounds than typical for their age, speaking late for their age, often leaving out sounds when speaking, and using speech that is hard to understand. Symptoms of dysarthria include slurred or slow speech, not being able to speak louder than a whisper, or rapid speech that is difficult to understand. There are some therapy options for childhood apraxia, like speech-language therapy, and that includes evidence-based methods such as rapid syllable transition training and dynamic temporal and tactile cueing. These methods help retrain the brain and improve the coordination between thought, language planning, and muscle movement. Because this gene influences motor pathways and timing, therapy often focuses on repetition, rhythm, and building new motor patterns. And even though FOXP2 is a genetic condition, therapy can create new neural connections that help children improve significantly over time. And I chose this gene and phenotype specifically because I've spent a lot of time in schools with young children, and seeing their speech therapy sessions has always been really interesting to me. It can also be very emotional, and seeing how excited they get when they improve with their speech is pretty exciting for me. So I thought this would be a good opportunity to learn more about the science behind certain speech disorders. And understanding the biological side kind of adds a new meaning to it. It's not just what the symptoms are, but why they happen, and how genetics and brain development play a role. Moving on to the location of the gene, as well as some information on the alleles. My research is focused on the human-specific allele, although there are many documented variants of the allele in humans and other species, and these variants slightly change the FOXP2 protein structure. Even small changes to this allele can affect how the protein functions, which can influence speech abilities. For transmission of the altered gene, only one altered allele is necessary to pass it down to offspring, but I will get more into that later. This gene is located on the long arm of chromosome 7, specifically within the band 7q31.1, and it has about 603,000 base pairs. The image on this slide shows the location of the gene, and you can see chromosome 7 and within that the band that marks where FOXP2 is found. And this location in particular is important because other genes in this region are also involved in brain development, so deletions or rearrangements in this area can sometimes affect multiple traits at once. Moving on to transmission patterns and gene expression. The FOXP2 gene is transcribed into mRNA, which is then translated into the FOXP2 protein. That protein then enters the nucleus and binds to specific DNA sequences in other genes, either activating or repressing them, like I touched on earlier. This action shapes neural circuits necessary for speech and language. In other words, when FOXP2 activates the right genes at the right time, the brain develops the pathways it needs for planning and producing speech. When this gene is expressed, it's expressed in several parts of the brain, including the basal ganglia, which is involved in planning and executing movement, the prefrontal cortex, which is involved in higher level motor and cognitive control, and the cerebellum, which involves fine-tuning movement and timing. And this image at the bottom shows exactly where these parts of the brain are. Next, I'm going to talk about the relationship between this gene and Mendelian principles. The FOXP2 gene is located on chromosome 7, so it's an autosome, and an example of how Mendelian principles operate in humans, because each parent carries two alleles for this gene, and the law of segregation states that these two alleles separate during gamete formation so that each gamete receives only one allele. Consequently, a child inherits one copy of the FOXP2 gene from the mother and one from the father. When an organism inherits two different versions of alleles of a gene, the dominant allele determines the physical trait or phenotype, while the recessive allele is masked or hidden. FOXP2 also demonstrates a dominant inheritance pattern. This means that inheriting a single altered copy of the gene from either parent is sufficient enough to cause a speech or language disorder. But this gives families a very clear pattern that affected individuals appear in every generation and have a 50% chance of passing the altered gene to their children, which is an example of the law of dominance. And at the bottom here, I have an example Punnett square that illustrates the possible combinations of alleles, showing the genotype ratio of 50% heterozygous and 50% homozygous, and the phenotype ratio of a 50% chance of an offspring being affected. And this helps us visualize what's happening genetically. Lastly, I have a pedigree of the FOXP2 gene that shows the inheritance of the gene through a few generations, and in this example, the affected female has passed down an affected allele, and in this example, the females have been affected, but anybody can be affected by this gene. And you can see how the disorder moves through the family, which matches the autosomal dominant pattern that I discussed earlier. Pedigrees like this are a great tool for understanding things like family history. And these are my sources.